Welcome to on-demand training for FlowCam 8000. In this video, we will describe the workflow for performing flow imaging microscopy with FlowCam. The video is presented in 3 parts. In the first part, we will describe how to autofocus the FlowCam 8000 optical system using standard beads. Next, we will describe how to establish custom context settings using standard beads just for demonstration purposes. And lastly, we will describe how to run samples on your FlowCam 8000 using auto image mode. The first thing you will want to do when performing flow imaging microscopy is to make sure the optical system is in focus. Performing autofocus can be done directly from the camera view of visual spreadsheet. To start, click on the camera view button to open the setup and focus mode window and the pump control window. From this view, you can inspect the camera view and confirm that the black lines marking the outer edges of the flow cell's inner channel are properly aligned with the camera's acceptable region, which is marked by the red lined border. After placing a pipette tip into the sample port and adding approximately 100 microliters of water, you can click on the autofocus button. The autofocus assistant will welcome you. Clicking Next takes you to the confirmation dialog asking you to check if the configurations, as presented in the configuration status bar, are correct. If the configurations are not correct, cancel out of the autofocus routine and reconfigure the hardware accordingly. Otherwise, click Next. The autofocus assistant will then ask you to select the autofocus algorithm to be used from 2 options: best bead count and best focus image. The best bead count algorithm finds the focus position that achieves the highest average image quality across the entire particle population. We suggest choosing best bead count if, for example, you are primarily interested in attaining accurate particle count and concentration values and are satisfied with average overall image quality. The best focus image algorithm finds the focus position that achieves the highest peak image quality for a subset of the population. If you are interested in attaining maximal quality, high-resolution images of a subset of particles in your sample at the expense of particle count accuracy, we suggest choosing Best Focus Image. Best Focus Image, for example, may be desirable for producing journal-ready reference images. Best Bead Count is desirable for most applications. However, which algorithm you choose will ultimately depend on your particle analysis goals. We recommend trying both to determine which works best for you. Next, you will be prompted to add focus beads to the sample port. We recommend adding 4 to 6 drops of the standard beads that come with your instrument and can be found in the supplies and consumables box. Note, different size beads should be used for the various objective lenses according to this table. You can find this table in the FlowCam 8000 user guide in your desktop resources folder. Once 4 to 6 drops of beads are added, fill the pipette tip to 1 milliliter with ultra-pure water. If you would like the beads to be automatically primed into the flow cell as part of the autofocus routine, be sure the tick box is selected. If left unselected, be sure that the focus beads have already been primed into the flow cell before the autofocus routine begins. Clicking Next, the priming pump dialog appears indicating the initialization of the pump syringe. You will see the beads enter the flow cell from the camera view window. The autofocus assistant dialog asks you again to confirm the configuration settings. This checkpoint is an opportunity for you to see that the focus beads have sufficiently primed into the flow cell. If everything appears correct, select Finish. During the autofocus routine, the focusing dialog appears and the collage window will open alongside the camera view window. You can arrange these windows to optimize your viewing. Images of individual beads will populate the collage window in real time as they are segmented from the full field of view camera images. The segmentation of individual particles from the background camera image is identified with a red box around each particle. You will notice the change in focus quality as the linear stage adjusts the objective lens through the various focus positions. When the autofocus is complete, a dialog will appear indicating success. Click OK and go back to camera view so you can clear the flow cell. To speed the process, you may enter a faster flow rate as long as you do not exceed the maximum flow rate for your FOV flow cell as shown in this table. Select Start Pump and let the focus sample clear from the flow cell. When the flow cell is empty, complete at least one clean and rinse cycle and make sure the flow cell is visually clear and free of any particles. It may happen that focus is not achieved at the end of the autofocus routine. This typically occurs when not enough particles were present in the focus solution. Click OK, rinse the flow cell, and after checking that there are sufficient beads in the focus solution, repeat the autofocus protocol. With the FlowCam optical system in focus, you may now establish custom context settings for your specific sample. Select the Context button from the Visual Spreadsheet operation bar to open the context window. From the Load tab, you can choose to load a factory default context file or load a previously saved custom context file. When you click on the Load a Context File button, the Open Context window opens, listing the names of saved context files you can load from the database. The Load Context dialog appears, confirming that the selected file successfully loaded. When you click OK, you will see the name of the active context file in the title bar of the context window. The Capture tab allows you to input parameters that will help Visual Spreadsheet discriminate discrete particles during camera capture. There are 5 panels in the Capture tab: Particle Capture, Particles Defined by, Images, Acceptable Region, and Rolling Calibration. In the Particle Capture panel, you can input a value in the Distance to Nearest Neighbor field to tell the software how far in microns particles need to be from each other to be considered distinct. The Close Holes Iterations function is used to fill in small gaps, or close holes, in an image. Choosing a value of 1 through 5 defines how many times this morphological operation will be repeated using a 3x3 pixel neighborhood with each iteration. In the Particles Defined by panel, segmentation threshold conditions are selected. Depending on the particle type in your sample, you will want either just dark pixels, just light pixels, or both dark and light pixels to be used to discriminate particles from the background of the camera image. For example, with opaque particles, only dark pixels would be selected, while for samples with both opaque and translucent regions, like biological cells or aquatic organisms, both dark and light pixels would be selected. Recommended values for common aquatic and biopharma particle types are provided in the FlowCam 8000 configuration guide found in your desktop resources folder. The Images panel allows you to define the number of pixels separating the edge of the imaged particle from the background. You can also choose Save Raw Images and input the maximum number of images to save. The saved raw images can then be used in file processing mode. In the Acceptable Region panel, the position of the flow cell can be adjusted by changing the values in the left, right, and top and bottom fields. If changes are made here, it is good practice to go to camera view to make sure that the flow cell is indeed properly aligned with the camera's acceptable region. When rolling calibration is enabled, the background image is constantly updated throughout the flow imaging run to optimize the segmentation function. This is especially helpful with samples that tend to have sticky particles. With the rolling calibration function enabled, particles that stay in the camera view will get treated as background. Next, go to the Flow Cell tab. Confirm that the flow cell type shown in the flow cell panel matches what is shown in the configuration status bar. The Sample Settings tab contains 3 input panels: Settings, Estimated Efficiency and Runtime, and Priming. In the Settings panel, you can input values for the sample volume in milliliters, flow rate in milliliters per minute, and auto image rate in frames per second. Note that the syringe size shown in the Settings panel matches what is shown in the configuration status bar. Also check that the flow rate is set within the recommended flow rate range for the installed objective. The auto image rate and estimated efficiency values are related such that changing the auto image rate directly impacts efficiency. Due to the complex fluid dynamics of the flow imaging system, an auto image rate that does not exceed approximately 70% efficiency is recommended to ensure that repeat imaging of a given particle is avoided. Visual Spreadsheet will give a warning when the recommended efficiency level is exceeded. Recommended fluidic settings, including auto image rate and flow rates, can be found in the FlowCam 8000 series configuration guide in your desktop resources folder. In the priming panel, you can choose to have your sample automatically primed or manually primed into the flow cell at the start of a run. When automatic prime is selected, you can input a desired priming flow rate. This option is disabled when manual prime is selected, since the manual prime rate is set from the pump control dialog in the camera view. The Dilution tab allows you to input a dilution factor for Visual Spreadsheet to use in its particle properties calculations. In the Sample Dilution Concentration panel, when enabled, the software automatically calculates the dilution factor by finding the ratio between the sample volume and the total volume values that you enter. There are 2 panels in the Capture Filter tab for you to define conditions Visual Spreadsheet will use while acquiring images: the Basic Size Acquisition Filter and the Advanced Acquisition Filter. In the Basic Size Acquisition Filter panel, you can choose filters based on ESD, Estimated Spherical Diameter, or ABD, Area-Based Diameter, or opt to use No Size Filter. When using ESD or ABD, diameter minimum or maximum values can be entered. Advanced acquisition filters can also be selected from previously saved program files. The Reports tab has 2 panels, the Export panel and the Classification panel, which allow you to define how you want run data to be exported and how you want to manage classifications when the run terminates. The Find button allows you to access a classification from a saved location. The Notes tab presents a convenient space for you to jot information pertinent to the context file. The Stop tab presents a Stop Conditions panel and Recalibration panel. The Stop Conditions panel contains a list of options for stopping a run. In this demonstration, we are choosing to have the run stop automatically when either the sample volume is fully aspirated or the operator manually terminates the analysis. The Recalibration panel allows you to define specific time intervals for Visual Spreadsheet to recalibrate during a run. The Fluorescent Measurement Settings tab is only available for FlowCam 8000 instruments that are equipped with a laser. The fields available in the tab allow you to set the acquisition threshold, noise threshold multiplier, and the gain. Once you input your context settings in each of the tabs, be sure to go back to the Load tab and save any changes you made to the context file, or choose Save Context File As to create a new context file with a unique name. With the optical system in focus and the context conditions set, you are now ready to run samples in auto image mode. First, check the configuration status bar to confirm the hardware configuration and context file are correct. Then, click on the Context button to double-check everything looks okay. For example, note the correct context file name is in the context window title, that the appropriate flow cell size is selected, and the sample settings are consistent with your expectations, including that the proper syringe volume is shown. Click OK. Securely place a new, clean pipette tip in the sample port. Add the appropriate sample volume. In this demonstration, we are using a 500 microliter sample volume. Go back to Visual Spreadsheet and click on the Auto Image Mode button. In the Stop Conditions window, confirm the correct stop conditions are chosen and click OK. Enter a unique file name in the Run Name dialog box and click OK. The flow imaging microscopy run will begin with a brief calibration step, and then particle images will begin populating the collage window. With the Auto Image Mode camera view window and collage window side by side, you can watch how well the particles are streaming through the flow cell. It is also good practice to observe details in the run data text to get a real-time sense of how the run is going. When the run completes, the preferred particle data statistics and graphs that you have selected will automatically appear in the data display window. For a more detailed demonstration of how selections in the Preferences and Graphs menus are shown in the data display window, please watch the Layout Your Data Display video on the FlowCam University FlowCam 8000 homepage. Perform flow imaging microscopy is now complete.